let's give her a warm welcome, Dr. Vitalis.
All right, good morning. And thank you for being here on a Saturday morning. We're going to have some fun. So let's see, here we go. All right, I'm going to tell you just a little bit about how I became a scientist before I get into the actual science. First, I want to mention that this is a beautiful theater, and I'm very proud to be here and be a part of this event, and thank the organizers for inviting me, and of course, all of you for being here. So when I was about your age, the age of some of you, I was in school, I wanted to be a doctor. I love biology. I was fascinated by the human body, how it could change and adapt and heal. And I went off to college as a biology major on a path to become a doctor. And I loved the biology classes. I wasn't so thrilled with the chemistry. I didn't quite understand why these bonds were so important. And that was hard for me. I got to my junior year, took biochemistry, and just loved it. And it all came together. I understood why these molecules were so important. And then I wanted to learn more about how the body worked. I realized that was really what underlied how our bodies were working. And so I changed my major to chemistry my last year, and spent the year in the science lab, in the chemistry lab, and then went off to graduate school in biomedical science to be a scientist. And then I also wanted to teach, which I'm doing now as well at the community college. So I guess the lesson is you can always change your mind. You don't need to know right now what you want to do, but follow your passion. That's the most important. So with that, I'll start in here. And my talk is Menacing Microbes. Protein models reveal secrets. So I will be talking about some work that I do at the laboratory. Working at Lawrence Livermore Laboratory is wonderful. We have a multidisciplinary team. So as a biologist, I work with computer scientists, engineers, and everybody is important at the lab. Okay, start with some pictures. And I'm going to go through and show you some of these pictures and think about what they may have in common as I'm going through these. Okay, first of all, Streptococcus pneumoniae causes sore throats. It's a bacteria. SARS virus, relatively new, about 10 years ago, is making people sick. Hadn't been seen before, but similar to other viruses, it had these spike proteins that enabled it to grab onto its host. All right, hepatitis virus exists in several different forms. Influenza can be quite devious as it changes its form. That's why we need a new vaccine every year. Salmonella, bacteria, it can cause food poisoning, can make us sick. Staphylococcus aureus, it's on all of us. Usually doesn't make us sick, but it can become menacing and cause skin infections. E. coli bacteria, lactobacillus bacteria, it's in all of us, in our digestive tract. We need it to be healthy. It's a friendly bacteria. But rabies virus, this is not so friendly, and we don't want to encounter that one. And here is Ebola virus. As we know, this can also be deadly. It can circulate in the bat population or other animals and rarely enters the human population, but as we all know, it recently has. So we want to understand what it is about these microorganisms, how they can actually change. Okay, so have you been thinking about what they all have in common? They're all microbes, tiny living organisms, and then we're going to discuss some other aspects, how they're common. Okay, but we'll look at some more examples and how we study them. Okay, Yersinia pestis is a bacteria. Back in the 1300s, is particularly deadly. About half the population of Europe was coming down sick and dying from this bacteria over the course of about 4 years. And the people were very frightened. They didn't know what was causing this disease, how it was being spread. They couldn't see it. So, without knowing the enemy, that was very frightening. However, a few hundred years later, microscopes are developed, and scientists were able to see these microorganisms, and soon they made the link with diseases. And a particularly groundbreaking discovery was made by this scientist on the right, who you may have heard about, Alexander Fleming. Several years ago. Well, he went on vacation, but he came back and he noticed that he had left a petri dish sitting out by the window and had captured some mold. And he noticed that there was a clear spot next to the mold where the bacteria had died. And he then studied this mold for potential to kill bacteria, and antibiotics were developed. Sulfa was the first one that came to market, and then penicillin. And this was a miracle drug. In fact, the cover of Life magazine back in the 1940s, medical miracle. The scientists had conquered deadly infections. And it was truly a miracle. As the soldiers are coming back from World War II, they were no longer dying from gangrene and infections. Life expectancy was greater, and everybody was happy. The deadly bacteria had been conquered. Or had they? What was going on was eventually the drug quit working. The antibiotics were not working because the bacteria can change. They can adapt to become resistant to the antibiotics. So this is important for scientists and doctors to understand. And they really want to know, how can they change? How can they actually adapt to become more menacing? Okay, so we want to think about, how can we study this? How are they changing? And so let's go back to, what do they all have in common? So this is our first question of the morning. I know it's a Saturday morning, so this question, and I'm going to call out some answers, and you raise your hand when I call out an accurate answer. Okay, what do they have in common? Do they grow? All right. Do they change, adapt to survive? Do they have DNA? Do they have proteins? If you raise your hand 4 times, you're right. 4 times already this morning. That's really good for this early in the morning. So, okay, let's look at these aspects first. DNA, that is in all cells. Here we have, it's the green inside this bacterial cell. DNA is very similar, though, in all living organisms. We have trillions of cells, but they all have DNA that's very similar. And it is important because it carries the information to make proteins. Okay, why are proteins important? They come in thousands of different forms. They have unique shapes, and they carry out all the important roles of the cell. They enable the cell to eat, to reproduce, to grow, to communicate, and to survive. And so what's really a mystery, though, is how can all these proteins carry out so many different roles? Well, it's really because they have unique shapes, and their shape determines what their function will be. So that's a key to understanding how microorganisms work. And this is such a fascinating process. I know some of you are learning this in class, or you've just covered this. So I'm going to show a little video clip that'll show it live. It's really phenomenal. There's some details that you may learn in future biology classes. Okay, we won't learn all the answers today, but that shows you how complex the process is, and those events are occurring in the cell. But we have a mystery here. Now we have this string, this wiggly line. That's a string of molecules called amino acids that make up a protein. But we want to understand that this is just a straight line. How does this protein take on this complex shape so that it can carry out its roles? And in order to do that, we want to understand the interactions between these amino acids. There's 20 different amino acids, but they have these different chemical properties. And I have a demonstration. Are there 15 of you that would want to participate? Or we can try it? There are 15 of you who want to be, we want to create a human protein. It's 15 amino acids, and if not quite that many, we can pare it down. But this will be fun, because then you can teach all of us how this protein chain of amino acids is actually going to become a complex protein that can carry out a role. Okay? So as you come up, you're going to line up in this order, but we cut out a couple of the amino acids so it wouldn't be quite such a long chain. So how about the methionine? Whoever has the yellow methionine be at this end. Yep, and then line up behind lysine. Follow, okay, yeah. Follow, let's see. Face him. Face him.
Oh,
you face this way. Everyone, we're all going to face this way.
Face.
Yeah, everyone face this way in a long train.
I'm
Yep.
sorry, face this way?
Yep, face
Okay,
this way.
okay.
Yep, you guys are great. And hold your card up to the left, and then put your right arm, if this is okay, on the shoulder of the person in front of you. Okay. So we're going to be this amino acid chain. All right, this is great. Okay, so I'm going to back up real quick here and say these amino acids each have a different chemical property. And we're going to see how this chemical property is going to enable this string of amino acids. You guys are great. But we want to see how this protein can do something special. Okay, so the cysteines, the cysteines are going to want to form a disulfide bond. So they're going to come together. So the two that are green, raise your card, the two cysteines, you're special. You're going to come together, but something has to happen. You're all stuck together because you're a chain. So as the cysteine at that end is going to wrap around to get close to this cysteine, so you're going to wrap around, or you can come this way. Actually, come this way. This is great. But you all have, you have to be, you're all connected. Yep. So the glycine, yep, you still have to be, yep. Yeah, so these two cysteines are going to get together. Oh, see, now we already have a protein forming. This is great. Negative and positive want to get together. Okay, so where's our negative white and our positive red? They're going to want to get together. Where's the red and the other? Oh, they're gone. Okay. You know what? I'm going to switch you guys around. You're going to be the glutamic acid, and you're going to be, okay. This red and this white, they want to get together too because it's a positive and a negative. We just changed our protein chain a little bit. Okay, this is great. Now something else happens. The hydrophobic, they want to get away from the outside of the cell, so they're going to kind of get close together. So the yellows are going to kind of move in a little bit. And then the blues, that you want to get away. You're hydrophilic. You like water, so you're going to move away from the other amino acids. So you kind of pull away. All right? So now look at this, but you're still hanging on to each other because you're formed by a peptide chain. So look at this protein. Isn't this cool? They started out as a long chain, but now they are something really special. They have this shape that was determined by the chemical properties of the amino acids. So great job. You guys are a wonderful protein. Thank you. And I'm sure that you're doing a very important role in the cell. Okay. All right. Without you, we couldn't visualize how this protein took a shape either. So, terrific. Okay, so this is what you were able to demonstrate, and you did a fantastic job, that we started out with this string of amino acids. That was like the green string that you may have caught in the movie that came off the ribosome. And it folds up. Those cysteine bonds are very important for the shape of the protein. So, a little story about how cysteine bonds are important. Okay, so this woman was bored with her hair. She went to the salon, and they broke all the cysteine bonds in her hair with a chemical. Now, they're made of sulfur, so that's why salons kind of stink sometimes. And then they reformed those cysteine bonds in a different way. Now she has a new look that just shows hair is made of protein like the rest of us. Okay, sometimes proteins can have a very small change. If one of those amino acids was different, the protein would fold different and it would have a different function. So we all love cats. Siamese cats are special. They have an enzyme called tyrosinase. Well, they all have tyrosinase, but theirs has a mutation so that the melanin production from tyrosine does not happen when the cat is at its core temperature. So melanin is only produced where it's not close to where it's warm. So this cat is from a warm climate, probably California. So only the very extreme parts of the body have produced melanin, are dark. This cat is probably from the upper East Coast where we had all the cold this year. Okay, so he has more melanin. So the key is that this enzyme functions differently, and it's really just an amino acid difference. Well, we all love our furry friends regardless of their color, so this isn't a problem. However, there are situations where there's a one amino acid change that can cause a change in the function that can have a dramatic and devastating effect. An example here is a molecule called hemoglobin. They're tiny little molecules in our red blood cells that carry oxygen. And there's a particular amino acid that normally has a negative charge, but it can become mutated and it becomes hydrophilic. Okay, negatives, raise your hand. Oh, you took them away. That's okay. Okay, remember the negative, then he scooted over to get close to the positive, so that was important for how that protein, how it took its shape. Well, that doesn't happen here, and these red blood cells do not carry oxygen very well, and it causes sickle cell anemia. Okay, so to understand function, we want to look at how proteins, their sequence, how a mutation can change their shape and their function. And scientists are interested in this in microorganisms as well, because that's a threat to human health, right? So how might we, if we want to understand how microorganisms are going to be changing and adapting to make us sick, what might we look at? Anyone want to venture a guess based on what we talked about? What part of the microorganism? It's probably such an obvious answer that says the proteins. Yes, we want to look at the proteins and how their sequence, if their amino acid changes, how it can affect the structure and the function. But we have a challenge. Proteins are small. We cannot even see them under a microscope. All right, so we need to develop a way to study them. And here at Lawrence Livermore Lab, we use models to study molecules that we can't see or feel or touch. That's how we can explore them. Let's build a model. Okay, in the case of protein, what we start with, we just have a sequence of letters. So each of these represents an amino acid. So these are not nearly as interesting as our human amino acids that were up here. But this is the sequence. It's not very informative as it looks, but what our scientists at the lab have done is they've built a tool where they can actually build a model based on the sequence. So computer scientists and biologists have been working together to develop a tool where they take advantage of all this structural information that's available. So the structure of a protein, I'll give just a high-level description of that, can be determined using crystallography or nuclear magnetic resonance, some very complex science methods. But that data is available to all scientists. Scientists share their data. So computer scientists take advantage of all the data that's available to be able to create their own model. And that's the power of computations. Homology means like, so the protein is compared against all protein structures that have a similar sequence. Nature repeats itself, so if we want a protein that has a similar role in different organisms will look similar. So we can take advantage of that. And then with this we're able to use this system, and we build a model. So now this actually represents the sequence, but with this we can look at it and predict if we change one amino acid, how will the structure change? How might the function change? And if it's in a microorganism, how might this make it more menacing, right? Okay, so here's one example where we use this tool. Yersinia pestis, again, a collaborator had a strain, and he observed a mutation, and he saw that these bacteria were growing faster. So he sent the sequence to us, and it was in this region of this long, complex protein. I'm going to drill into the interesting part. That's what scientists can do, is computer scientists can really focus on what's of interest. There was a hydrophobic amino acid normally at this location, but what they found that it was mutated, okay? And so they wanted to look closer, this mutation of interest. In the normal protein, there was this tunnel here, which they determined was important for the function of the protein. Here's where there was that hydrophobic amino acid. However, the mutated one, it was positive. And remember how our positive amino acid kind of scooted over to get next to the negative one? That happens here. It closed the tunnel, and then this protein was predicted to no longer function. So using these models and what we know about amino acids, we made this prediction. And this is really important to help us understand why these bacteria may mutate faster. Okay, so let's not ignore the viruses. Those actually mutate much faster than bacteria usually. And it's very concerning if a virus that is present only in animals can adapt a mutation to make it capable of infecting humans. With influenza, for example, swine flu, and then now we all know with Ebola, if it can actually mutate so that humans can get sick. That's of concern. So a couple years ago, some scientists at the lab did some experiments where they used a bovine coronavirus, which normally only infects cows. So this cow has this virus. He's probably not very healthy. And it's similar to SARS, so they're interested in this spike protein. They took some of those cells and they grew them in cells that were from humans, not in the humans themselves, but they get cells and they grow them in place. That's how scientists do the work. They wanted to see what were the changes that might make this virus actually adapt to humans, because that would give a clue how other viruses might make that jump. It's called host switching. So that's important to understand how these viruses might become more menacing that way. So what they did was this is the sequence, and these are the mutations they identified in the cells that then grew in humans, the strains that grew in human cells. They mapped, this isn't very interesting itself, so our computational scientists mapped it onto a protein here, and these green represent these green amino acids. And he saw, this is the spike protein, he saw they're on the surface, and that was an important clue to how this protein could change its shape and better enable it to infect the humans. So this is a start towards understanding how viruses change. But it's looking at the proteins and how they change and how that changes their shape. Okay, so that's another example of the types of experiments. And this is an example of biologists and computer scientists working closely together as well. Okay, I want to go back to the bacteria because they're very pesky and they can change. And we saw how they can change so that they no longer are responsive to the antibiotics. And this is another area we study at the lab. So I'm going to show another video clip here to show bacteria have been very menacing in that they have several strategies to evolve and adapt so that the antibiotic drugs no longer work. So this video is going to show some of them, and then I'll describe one of the mechanisms in another example. Okay.
Gram-negative bacteria have a cell wall that lies between the outer membrane and the inner cytoplasmic membrane. The cell wall contains penicillin-binding proteins, or PBPs. PBPs help build new cell wall during growth and division, maintaining its integrity. Beta-lactam antibiotics bind to these PBPs and weaken the cell wall, resulting in the cell's death. Gram-negative bacteria, common in hospital-acquired infections, often contain several mechanisms that can render antibiotics ineffective, including changing the outer membrane and preventing antibiotics from entering, and pushing antibiotics out that do get through by using efflux pumps. Certain gram-negative bacteria can also produce beta-lactamase. These enzymes inactivate beta-lactam antibiotics and keep them from binding to PBPs. Some gram-negative bacteria can produce powerful beta-lactamases called extended-spectrum beta-lactamases, or ESBLs. ESBLs can render penicillins, cephalosporins, and monobactams inactive. In order to effectively treat infections caused by drug-resistant gram-negative bacteria, new antibiotics that overcome multiple mechanisms of resistance are needed.
Okay, so those beta-lactamases, those were key. That's one particular enzyme that bacteria are starting to produce so they can break down the cell wall. And so that was the one that was highlighted. And about 4 years ago, we were alerted by some collaborators that there was an outbreak of a new potential superbug bacteria that was creating this beta-lactamase called New Delhi metallo-beta-lactamase. New Delhi is where the outbreak began, but it spread rapidly, and what was frightening was the bacteria that carried this gene were resistant to all of the known drugs. And they were interested in our collaborators and scientists wanted to know what was the change that was making it resistant, how had it adapted. So the collaborators sent us some sequence of this new protein of the NDM1. What do you think we did with the sequence? We built a model. Is that what you said? Build a protein model. Yes, that's exactly what we did because we had the tools to do it, and that would help us to predict how that change might make this new gene, or the protein made from the gene, how it would make these bugs so resistant to the antibiotics. And the structural analysis that was carried out, this is what the protein looks like. In green, and this is a little bit advanced, but I thought I'd show you a little bit of the complexities. Areas in green are unique to just the NDM1. The areas in red are similar to all of the beta-lactamases. So we know that those are important for the function of the protein. But there was this discovery that this protein had this extra loop, and that might be important for how it worked. And it also had some amino acids in the critical active region of the protein that were strictly unique to this new NDM1. So we handed this information back to our collaborators so they could study it to understand how this bacteria was evading the drugs and also to help them develop new drugs that were more powerful. But this might all sound discouraging that the bacteria just keep evolving away from the drugs that we have available, and viruses also evolve, so they become more menacing. So I'm going to show the little bit advanced model. So this is our NDM1, and our computer scientist has enabled the 3D model so we can really get a good look at it from all different angles. Proteins are three-dimensional. So in the tools that we use, we can help to try to develop new antibiotics. So there's a new area of research that's quite exciting where we're actually taking advantage of the bacteria's own mechanism to destroy the cell wall and try to develop drugs that we can use as novel antibiotics. But the fight is still out there, so we need new scientists and all you bright scientists that will be doing research in the next few years to help us develop new drugs. So we're always a concern with the menacing microbes. So I'm going to just make a quick connection here to the movie, which, as you may know, is a virus focused on a virus that propagates rapidly. And some of these concepts will be underlying the theme of the movie. I just want to add, because we were talking about antibiotic drugs. In regards to viruses, antibiotics are not effective, but there are drugs, treatments, potential cures, but I don't quite use the word cure because it's not always entirely effective. But different antiviral medicines, they need to be specific to the organism itself. They may block replication of the virus in the host or bind and destroy it. Here, I'll come over here. And then another line are vaccines. And this is really the hero of cures for viruses, in that they provide immunity, but they actually stimulate the body's own immune defense against the pathogen. Okay, so you'll see some of this in the movie, and it's a fantastic movie. So I hope I'm sure you'll all enjoy that movie. Okay, so I'm just going to summarize what we went over, and then we can, or what we've learned, and then we can have some questions. So all living organisms can change. All right, and this change by microbes makes them more menacing. So we want to understand how they change. Proteins, we saw, are really the clue. That's how living organisms can adapt and change. Okay, proteins, as you got up here and demonstrate, they fold into precise shapes, and that's what determines their function. And since they're smaller than can be seen, we develop models so that we can look at them, analyze them, and predict how they may change. Okay, and in doing so, we identify clues as to how these organisms can become more menacing. All right, so that's the end of the lecture. Do you have any questions? Yes.
So, from the discovery of a new superbug or whatever, to finding a cure or an answer or a way to battle, what is that timeline? Months?
That's a good question. It can be years. There's a large number of steps. There's the basic science, which is the kind of where we're at with this drug that I mentioned, and then developing the drug, and then clinical trials can take several more years. So it is a long process, and unfortunately with antibiotics, there have not been very many new ones in the last few years. But
relate that directly to Ebola, what's happening.
Okay, I can say a little bit. We have been able to, or what some of our team has done, and I didn't present it because we don't have any of this published, of course, try to understand how the virus might evolve and change so that some of the existing drugs may no longer be effective. And there are some that are actually available, antibody-based. I mentioned some treatments that actually can bind the virus, and that others that may stop replicating. But some of them are in initial stages, which you probably have read that they're trying to speed up the clinical trials. But yes, and then there's also this, the potential the virus may mutate, and then the drugs won't be as effective. So that's why we need to continue the science, even when there's not an outbreak, because the ongoing science gives the information so that when there is an outbreak, we have the information, and the drugs are in process. Okay. All right, so in the front row here.
How does a vaccination prevent you from contracting a virus?
Very good question. So vaccination is actually challenging the immune system with a form of that particular pathogen, maybe an inactivated form or just a protein from it. So your immune system thinks that it's been infected, and it'll create all the antibodies it needs to destroy it. And then there's memory in our immune systems. It's amazing. So even years later, if we're infected with the real pathogen, our immune system is ready to charge and destroy. So it's not a drug itself, but it's stimulating our own defenses. That's why vaccines are so useful. Yeah. Yeah.
I'm interested in protein modeling. So you're using known sequences, and you already know the structure of most of those proteins that those sequences go with, right? How close are you getting to using computers to model a sequence that maybe is unknown? Like you could put in a string of amino acids.
Yeah. It's called de novo modeling. It is challenging. There are steps towards doing that. The homology modeling is what I showed, where we have models that exist, and some of them are not as similar. So it is predictive. There are steps towards it, but it is more challenging. And I don't know if that quite answers the full question, but.
You've got supercomputers at the lab, and I was wondering if they were making moves towards trying to get really good at writing algorithms and trying to figure out. There's some work there, and most of what I've been involved with is the homology modeling, so the de novo. But that's a really good point that we base it on what is already known for these tools, and that is an area where computational capabilities can help. Yeah, you have a question?
Yeah, when you said that the bacteria and the virus is changing and that, I'm assuming that you mean that from generation to generation, like kind of a survival, or do they actually change in their own little life cycle? Yeah.
Oh, I see. No, it's as they mutate. Yeah, okay. Okay, I'll repeat the question. Yes, that's a really good question. Does an individual bacteria change and during its lifetime take on new function? And the microorganisms replicate very quickly. So it's during the replication process that the mistakes are made in DNA replication, and that's the basis of the mutations. So that's the primary. Yeah, so it's going to be the next generation. And so it is survival of the fittest, which is how they become antibiotic resistant. So if we can stop the bugs, the organisms from replicating, then we can stop the mutations as well. That's a good question. Yes?
Good question. What are your thoughts on the notion that people talk about how it's not always good to take antibiotics to allow your body to fight off viruses itself? Because what you were talking about, how viruses grow and adapt. These people say that you do more antibiotics
Yes.
It fights off the virus, and the virus, like you said, mutates and learns to fight off, and then it gets bigger and worse sometimes.
Yes, that's a very good point. So is there a reason to avoid taking excessive antibiotics? Because can that actually promote? Yes, and one, you brought up one good point. So viruses do not respond to actual antibiotics. Antiviral medications are used. Antibiotics, but they sometimes are used for viral infections, and they do not. They're not helpful. And antibiotics do, because of survival of the fittest, which is what you alluded to, the bacteria will try to mutate to overcome so that drug will not be effective against it. So that's why doctors say take all of it so that these resistant bacteria don't have a chance to emerge. But the more times that the bacteria are exposed to that drug, they have more opportunity to evolve, and then that strain of bacteria can pass to someone else with those mutations that can make it resistant to that.
It's kind of a case-by-case basis. Sometimes it is good to use antibiotics right away, and other times it's good to just hold
Yeah.
it.
If antibiotics are needed, they can be effective, but if they're used excessively or not finish the course or used when they're not needed, then it can promote more antibiotic-resistant bacteria to be circulating. So it's a very delicate balance, and it's challenging for doctors. It requires communication, too, amongst doctors and scientists. So, do you have another question? Yeah.
I know you guys have a lot of challenges with developing antiviral drugs and bacterial drugs. Is there any room for the importance of really targeting that particular bacteria or virus? Because I know like a lot of antibiotics, they would kill multiple bacteria, even your good bacteria.
That's right. Have it targeted towards the deadly bacteria. Yeah, that is one area, and that would make them more beneficial if they're not killing the friendly, because there are friendly bacteria in the cell. And there is work there. Broad-spectrum drugs can kill all the bacteria, but they have more potential of promoting resistance. The challenge is when a person goes to the doctor is knowing exactly what strain they have been infected with. If they don't know, then they can't give a targeted drug. So diagnostics need to improve in order for the drugs to be more targeted. But that would be desired, that the drug would treat only the actual strain that person is sick with to prevent additional resistance. So that's a very good point. Yeah, and you have a question?
Yeah, actually, just a comment to your question. There's, I don't know if you do this kind of work at LLML, but there's a lot of work that's been done on bacteriophages. Those are viruses that are particular to bacterial strains. So that will actually address your question, because those bacteriophages are very specific to specific strains of bacteria. But that's a whole other realm.
And it is an area of research beyond just the lab, too, yes, and developing new antibiotics. Yeah. Excellent questions. Well, thank you very much for joining me today and for being here. Enjoy the movie. I don't know if Joanna has additional